Hi everyone, I'm Raphaël Lugan and this is a course on metabolomics. So here is the outline of this lesson. The first chapter is about definitions, defining metabolites and metabolism, and situate metabolomics within the wider omics approaches. Then I will give you basic lessons about analytical chemistry, including extraction techniques and chromatography. Then the third chapter concerns mass spectrometry, looking for the basic principles and then the different ionization sources and the different mass analyzers. The fourth chapter is about data analysis. This is a big chunk with first the cleaning of the data, tracking errors before working, then the raw treatment of the data, and finally the data interpretation, including a specific chapter about molecular structure and elucidation. And finally, a few examples of metabolomics application in different domains like foodomics. Well, I chose the domains that might interest you rather than me, in fact, but foodomics, functional genomics, and clinical diagnosis. I want first to, before starting, to specify that this is an introduction to metabolomics. We can't go deep into every aspect of it because we would need several, maybe 100 hours to cover everything, and I might not be able to deliver every information. This is oriented toward mass spectrometry metabolomics, but there is a whole range of application of metabolomics which are based upon NMR, nuclear magnetic resonance, instead of mass spectrometry. And there would be a lot to say about that also, but this is not at all my area of expertise. Concerning the data treatment, this is a huge limitation of metabolomics today, especially the alignment of the data and the reliability of the final data set, the refined data set, is still somehow limited. And another point is the molecular structure elucidation. This is a whole area of research and development and techniques, and we can't go too deep into this. So the idea is to give you a broad view of metabolomics with every aspect of it, and so that maybe if you do metabolomics one day, you have all the basis to really start digging into more specific processes of data analysis or even data acquisition. It takes months of practice to master a machine of a mass spectrometer, to really understand how to use the different parameters and what are the different problems, and there are many that can happen. So okay, so I want to limit the ambition of this course, but it's taking into account every aspect of metabolomics. So let's start with definitions to really know what we're talking about. And first of all, we need to define what are metabolites and metabolisms. So metabolisms could be defined as the set of biochemical reaction and transformation of organic molecules within a living organism. And the best way to show that is this little drawing with arrows and cycles, because this is how it looks like. You have, on one hand, you have the substrates on the left part, and on the right part, you have some products coming out of this. Metabolites are the small organic compounds which are indeed the substrate, intermediate, or products of metabolism. So they are represented here in blue. We call them small molecules because usually their mass is below 1,500 Dalton. An important notion is the metabolic pool, because this is the quantity of a metabolite accumulated in a defined biological compartment. In pharmacognosy, when people extract different substances from plants or fungi and so on, they usually use the whole organism or big parts of it, big chunks of it, like the roots or the leaves and so on. So in this case, the pool contains, well, the whole leaf. But we need to understand, for a biologist, it's very important to consider that metabolites are compartmentalized within the tissues and within the cells, even at the subcellular level. And so, for instance, while the concentration of caffeic acid in the vacuole of plant leaf is a different notion than the caffeic acid concentration in the whole leaf. So we need to remember that. Then comes the metabolic pathway. Well, it's a sequence of enzymatic reactions involved in metabolism. So here you have a linear pathway with a substrate on the left and a product on the right, and every step of transformation is catalyzed by a particular set of enzymes. Sometimes it's only one, sometimes you can have different enzymes producing the same reaction. Those enzymes, of course, are proteins encoded by genes. And so we can draw metabolic networks. It makes very beautiful posters on the wall of many labs. And this is a set of metabolic reactions, of enzymatic reaction, of metabolic pathways. And it makes a map because you have connections between the different parts of the metabolism. So in this case, you see you have here, well, just a very small subset of reactions. But you have the respiration, the photosynthesis, well, all the basic reactions in a photosynthetic plant tissue, and many central metabolites like the metabolisms of sugar, nucleotides, proteins, and amino acids, of course, and so on. And so you have here a metabolic map which can give you a topological, a static description of the pathways. It's everything that is theoretically possible in this organism given its genotype. But the limit of this type of representation is you have only a static view, and everything about the dynamic of this and when and why those reactions occur or not within a particular real organism is not addressed by this representation. So to give some notion about the extent of metabolism, we can consider a few figures. Like first, in plant kingdom, we estimate that there are between 50,000 and 200,000 of different metabolites. So it's a lot. It's a limited number, of course. It's not infinite. But in the biosphere, only in the plant kingdom, well, you have many structures possible. And then you have a typical eukaryotic cell. Well, I don't really know what it means, typical, but it's, once again, just to give you a notion about it. Well, in one cell type, you will find between 500 and 5,000 metabolites. So this is a range of this diversity of metabolites. We are used to classify metabolites into two categories. The first one is primary metabolism. By definition, it occurs in all plants, and many of them occur in all organisms, all known eukaryotic cells, for example. And well, these primary metabolites, they share biological function, some of them being universal in the living kingdom. I could give an example with photosynthesis, respiration, protein biosynthesis and degradation, membrane biosynthesis and degradation. So everything that concerns the basic functioning of the cells. Some examples of primary metabolites are sugars, that we call O's, organic acids, amino acids, nucleic acids, and fatty acids, and so on. Then comes the secondary metabolism. So it characterizes certain families, genera, or species. So by definition, we don't find them in every living organism or in every plant. They are associated to a particular genotype. And the main goal of these these metabolisms is to adapt to a particular ecosystem, to resist to a given pest or disease. It's an adaptation to a particular climate, whether it's dry or cold or wet. It's also about the competition with other plants within the same area, and it's about also the reproduction, the efficiency of reproduction, especially by communicating with insects in the higher plants. So in this area, we will find many toxic compounds, defense compounds, pigments, chemical messengers, and so on. So a few examples of secondary metabolites: the families of phenylpropanoids, betaines, terpenes, alkaloids. These are the main, but there are many other. We could cite cannabinoids and so on. Now let's talk about metabolomics and its integration in the omics techniques. When we talk about metabolomics, we think about different other words that are associated to it, like GCMS and LCMS, which are the techniques used to make metabolomics. But you will also listen lipidomics, secondary metabolites, primary metabolites. So these are subparts of metabolomics. Of course, the pathways, but also some approaches like untargeted approaches or fingerprinting, but also big data, because the techniques, the multivariate statistics used to analyze omic techniques and metabolomics are the same that can be used for big data analysis, and so on. So what is it really about? First, let's talk about the omics. The idea of the omics is to catch a whole level of organization within a cell. And the first term that was coined was genomics. The suffix omics means global, total. So it means that the genomic era was reached when the full sequence of the genes of an organism were available. So we catch the genes now very well. It's very easy to sequence and to reach the genome of a species. This is the whole set of its genes. It can be tens of thousands. In human, I think it's 24,000 genes. It's roughly the same in Arabidopsis, the model plant. So this is the range, the scale at which we can reach the genes today. We can catch them all, and so this is genomics. Then you have transcriptomics. The idea here is to quantify all the messenger RNA within a biological sample. And so it's based on the same techniques of sequencing, but you introduce another requirement, which is quantification. So the transcriptome is the abundance of every messenger RNA in a given sample. This is also something that is quite easy to do today. It's a mature technology, and you can really have the transcriptome of a biological sample. It's quite easy, but then you have to treat the data. It's another problem, but it's possible. The term proteomics is exactly the same, but with proteins, and it's much more difficult for a simple reason. Well, you can sequence proteins, but the difference with genes and messenger RNA is that you cannot amplify proteins. You can amplify gene sequences, you can amplify nucleotide sequences with PCR, polymerase chain reactions mainly, but you cannot amplify a set of protein. So the quantification is much more difficult, especially for low trace levels of proteins. And also the proteins are much more diverse physico-chemically than the nucleotides. Nucleotides are just nucleotides. It's 4 bases, and once you know how to analyze them, you can do that on any kind of gene. For proteins, it's much more complicated. First, because you have 20 amino acids, so the physico-chemical properties of proteins is much more diverse first. But then the proteins are modified. They can be glycosylated, phosphorylated, transformed in many ways. that will radically change their physicochemical properties. So it's much more difficult to keep this information and to find it. So proteomics do exist, but it's less efficient today than the other two omics techniques we mentioned. And finally, metabolomics. Well, it is the same idea. This is the idea to catch all, to quantify and identify all the metabolites in a biological sample. And, well, we try, but this is not at all the case today. It's an impossible task because mainly of the diversity of the physicochemical properties of metabolites. These omics techniques have been developed with the idea somehow to go toward a systems biology approach of biology. So we imagine that biosystems, this is a small drawing on the left, are a set of different levels of organization that are with a hierarchy and retroactions, of course, and that there is a flow of information that goes from the genome to the phenome. And of course, the different steps in between are the transcriptome, this is the expression of the gene, and then the proteome, which is the traduction of the transcriptome into actual proteins which will act physically on their cellular environment, and this will involve metabolites, and so the metabolome. And finally, this will result in the phenome, which is the full set of characters of an organism. So this is a bit simplistic because, of course, all this occurs in and interacts always with the environment. So this is something we'll talk about also later. So the idea is to catch as many information as possible on these different levels, and of course this was made possible because of the progresses in bioinformatics and just informatics, in fact, with the ambition to integrate this in models, in more and more complex models, and to understand better every mechanism at stake in a given environment, in a given cell. So how can we define metabolomics in this context? Well, the most ambitious definition of metabolomics is this. It is to quantify simultaneously, comprehensively, and in an unbiased way the metabolites in a sample. Well, there is no such thing in 2021. This is not the only definition of metabolomics. Many believe that metabolomics is an approach of untargeted nature, which means that you will try to cover as many metabolites as possible and just focus on the differences between two types of samples, maybe a control condition and then a treatment. And well, this is actually how we do it today. I will talk about that a bit later. But basically, the idea, if we take the same spirit, if we keep in the same spirit than for the other omics, this would be the definition of metabolomics. Try to have all the set and identify and quantify all the metabolites. Well, we saw the definition of metabolites. Well, these are small molecules occurring in metabolism, participating to metabolism. The simplest way to define metabolites is to say these are small molecules. So what does it mean, small molecules? How small? So, well, in general, it depends on the type of machinery and techniques used to analyze them. In GCMS, you cannot go above 1,000 Dalton. With LCMS, there is theoretically, with many machines, no limitation. So a few thousand Daltons is no problem. And in NMR, this is about the same. There is no real limitation. Then you have other limitations, which are the extraction of this compound, the preservation of the integrity, and so on. So this is to show you that, well, the limit between a small molecule and a macromolecule, we know what is a macromolecule. We call them proteins and nucleotides. But there are many compounds that are of high molecular mass, but are not really involved in what we call metabolism. We think when we think about metabolism, it's something that occurs fast and with high turnovers. Some of them are just storage for energy, like think about the molecule of starch. Some are just structures, like the lignin or the cellulose. Are they metabolites? Well, no, they are more macromolecules in a sense. But there is no conceptual definition of a metabolite. So you can have oligolignals that are metabolites, big sugars also. Small peptides can be considered as metabolites. They are not real proteins because they don't have any proteic activity. They act as signaling, like the cysteamine that we find in tomato. It's 18 amino acids, about 2,000 Dalton, and it's involved in the signalization of stress and signaling of stress. Well, it's a bit provoking, but small RNA could also be considered as metabolites, in a sense. Now, what are these analytical tools used to detect and quantify the compounds? Well, usually it's a combination of detection techniques aiming at quantifying and identifying the metabolites, and separation techniques aiming at simplifying a bit the profiles of complex extract. So the most common combination today is a combination of chromatography and mass spectrometry. With liquid chromatography combined to mass spectrometry, it's very common to analyze secondary metabolites, trace compounds, while when GC, which is gas chromatography hyphenated to mass spectrometry, we analyze other types of compounds like volatiles, fatty acids, primary metabolites. But we could add also some attempts to use capillary electrophoresis, which works for ionized compounds. Then the other big method is NMR, nuclear magnetic resonance. It has a lot of advantages because you can analyze any metabolites with this technique. The drawback is you need to concentrate them because there are limits of detections, and also there is a recovery of signals that make it more difficult to separate many compounds. So usually it's limited to a few dozens of compounds, but it's very powerful. It's a quantitative approach. You don't even need internal standards to quantify, as long as you know the structure of the molecule. And it's very fast. You can have analysis. You can analyze samples in something like 15 minutes or 10 minutes, depending on the goal of the analysis. And also it's non-destructive, so you can recover the sample and do other stuff with it. We have to mention also other detectors like the detector that can be hyphenated to LC or GC, which is flame ionization detector for FID for GC machines, and the spectrum analysis in the domain of ultraviolet or visible light, or the fluorescence also. But of course, this will be limited to the compounds that have these specific physicochemical properties. Whatever the techniques, the general design of a metabolomic experiment is the following. It starts with a careful sampling of organs, tissues, sometimes even organoids. What is really important at this step is the time scale and the kinetics that you want to cover. It's very important also to have an experimental design that was set up before sampling, of course, that will be useful and adapted to the type of statistics that you want to do, and depending on the kind of questions also you address. Then there is a step of extraction and possibly sometimes derivatization, especially in GCMS. There are many possibilities for extracting metabolites with solid liquid solutions, which is the widespread extraction technique, but also liquid. SP, you have to maybe adjust the pH and the polarity of the solvents So, well, we'll talk about that later. Then, of course, the step of separation and detection and quantification of the compounds, with mostly, we said, chromatography and mass spectrometry, but also NMR. And finally, the data mining and the data interpretation. And depending on the type of analysis, especially if it's targeted or untargeted, you might have different solutions. It's important to understand the limitation and the drawbacks at each of these steps. Upstream this workflow, you have compartmentalization loss. This is what I said earlier, talking about the leaf of a plant. If you analyze a compound in the leaves, of course, it has a very different meaning as compared to analyzing the same compound maybe in one cell type or in one tissue. You will have a dilution of the signal if you mix up different tissues with different concentration of the analyte. You have many sources of quantitative bias, and while the expertise means that you know the main traps you can fall in during these steps. And downstream the workflow, of course, depending on the choice of the apparatus, the machine available, and the kind of data analysis you will do, you will have problem of sensitivity, identification, limit of detection, quantification, some errors, and you have to use normalization, and to take a lot of to be very cautious to limit these issues. The main challenges concerning metabolomics are summarized here, and they are associated to some tough properties of metabolites. First, we said they are highly diversified. They are also present at very different concentrations. So there is a wide range of concentrations, and some metabolites have a significant activity at very low concentration. This is typically the case of hormones, in our case, phytohormones, while others are meaningful because they are very abundant. This is the case of glucose, which is the fuel, the basic fuel of every cell. Then the metabolome is highly dynamic. If you take so it's very important during sampling, and you have to take into account the natural cycles, like the circadian cycle. Metabolites content in an organism are changing constantly, depending on the environment, the moment of the day, the phenologic stage, the developmental stage of the organism. So it's changing, and this is something you need to take into account, especially during sampling, because after sampling, well, sampling is just basically a destruction of the organism. And so there is a reaction immediately in which some metabolites can be involved very, very fast. And finally, it's highly compartmentalized. We said that, and to answer efficiently some physiological question, it's often useful to know the compartment in which the metabolites are accumulated, and to know what kind of transportations of the metabolite occur in the tissues, between the tissues and between the organs. And this is something very difficult to address, especially at the low levels of distance. The smaller the compartment, the harder it is to access it. So as I said, it's almost impossible to make really a metabolomic well, in fact, it is impossible to make a metabolomic analysis today. So there is a strategy, and this is maybe the common current definition of metabolomics, which is very methodological. It means that you will try to find changes in metabolic profiles in different types of samples. So you have maybe a control plant, a wild type, and a transgenic plant. And then you look at the differences by doing an untargeted analysis, and you will focus on these differences by trying to identify the compound. And you won't even try to identify compounds that don't show any difference between these two types of samples. I say two types, it can be 3, 4, as many as you want. So we call them biological marker, biomarker, or metabolic markers. They are markers of a physiological difference, and this is what you study. So, well, and then the workflow is the same. So you need upstream a source of variation, like different genotypes, as I just said, like a wild type and a mutant. You also need, you can use also diseases and different treatments. And you can also use process in industrial in industry, like in food industry, when you take a plant sample, like a fruit, and you submit it to a different process, you will have metabolic changes. And then, of course, you sample them, you extract as many compounds as possible, or you target on a subset of compounds like secondary metabolites or primary metabolites, and then you look for the changes. We cannot understate the importance of bioinformatics and data treatment. So when you start your data treatment, you have raw information, and you have to transform that into knowledge. And so there are a lot of techniques that can be used, like mapping projections or multi-omic data integration, correlation network analysis, biostatistics, multivariate analysis. So this is very important to master at least the most common tools used to go from raw information to knowledge. So to underline these different approaches, I said non-targeted and targeted. We could add some intermediary situation with multi-targeted. And you can project that on this graph, where in APSIS you have the data quality in terms of identification and quantification. Of course, if you analyze one compound, the quality will be very high because you will use a method that is very specific and very sensitive and very efficient to analyze this compound. And then in ordinates, you have the number of metabolites addressed, analyzed at the same time. So in multi-targeted compounds, you can reach dozens or even 100 compounds, but they will belong to the same kind of family, to the same. They will have some basic common structure, like for example, they will all be amines or sugars or phenolic acids and so on. This is multi-targeted, so it's an intermediary situation where the identification and quantification is not that bad, and you can cover many compounds in a single run. And then you have non-targeted analysis, and this is what most people think about when they think about metabolomics. And you use one technique that has some advantages and some limitation for different types of compounds. And so you will cover hundreds of signals, hundreds maybe of compounds, but the quality of information will be much lower because you don't have pure standards to confirm the identity, and you will have many quantification bias that will need to be addressed. And so what we call metabolomics is mostly all these techniques together. And sometimes you do a targeted, sometimes you do non-targeted. You can go from non-targeted to targeted to confirm something with different philosophies. The non-targeted methods are very efficient at finding things you didn't expect. So it provokes serendipity. You observe, you make a new observation. And from this observation that is unexpected, you will have to generate a hypothesis to explain this observation. While with more targeted approaches, you will be, on the contrary, into testing this hypothesis. This is why you need to do both of them most of the time if you really want to solve a biological problem. It's important to mention also that the tools used are quite different. With non-targeted analysis, we will first compare the sample, then identify the biomarkers. The type of machine that can be used are LC-Q-TOF, Orbitraps, or GC-TOF, so mostly with high-resolution mass spectrometry. And then with multi-targeted or targeted, well, of course, you know what you target, so you know the compound. So you have a straightforward identification of them, and then you compare the samples. And the classical tool used in LCMS is a triple quadruple that is very efficient at doing this. So here are a few examples of multi-targeted analysis with a triple quad. It's named 3Q or TQ, triple quad. For example, a profiling of phenolic compounds. And so you have a chromatogram where every peak represents, obviously, hopefully, one molecule, sometimes several because you have coevolutions. The abscissa is the time of separation. It's a chromatographic time, and the ordinate is the abundance of the signal. So you can have a multi-targeted analysis of phytohormones. It's very useful to describe a plant sample. Then you can make non-targeted analysis with a GCTOF of primary metabolites. GCTOF, and after derivatization, I will explain that later, is a very efficient tool to profile sugars, organic acids, and amino acids. So mostly primary metabolites. And then non-targeted analysis in LCMS with UPLC QTOF. And in this case, it's the method is designed to cover mostly secondary metabolites with mid-polarity and mid-size. It is critical also to consider that metabolomics is a permanent method development. You are never done with one method that is okay forever. And if you just change your sample, or if you just want to add new compounds to your profiling, or if you want to improve the separation of a metabolite or another, you will have to make a method development. And this can be focused on extraction, like the choices for the choice of solvents, the temperature of extraction. If you have a new sample, well, you will have to do this development. If you have a new compound or a new experiment, it depends. It depends on how it has changed from the previous analysis. You can also want to check for the dynamic range and adapt the ratio sample mass to solvent volume during the extraction process to make sure that you are in the range of linear response depending on the concentration. Otherwise, you can miss some differences because, for instance, your detector is saturated, and so you won't see a doubling or a tripling of the quantity because you're already at the saturation level. So you'll have to do that for every new sample, new compound, and new experiment. The chromatography needs to be modified if you have a new sample, because at least to be checked, because you might have new coevolutions. If you look for new compound, usually you have to make a specific chromatography with proper pH and so on. Well, you always start from a basic method that works with most samples. But if you really want to improve your method and to make it very safe, then you will need to make a method development. The mass spectrometry follows the same logic. The sample exhaustion, you want to be sure that you have extracted all the metabolite in your sample. Otherwise, also, you will introduce a quantitative bias. So this will be very necessary if you have a new sample. If you made a method that works perfectly on peach tree leaves, well, and then you want to analyze a fruit of tomato for looking for the same molecules, let's say phytohormones, you will probably have to do this again, the sample exhaustion test. And then you will have to check for cross-contamination. Each sample and each method can bring its contamination. And especially one sample can contaminate the other, the following one that is analyzed in the machine, because not all the compounds have been eluted from the system. So this is also something very important to consider. It's a lot of work just to make sure your analytical method is adapted to the question you want to answer. One difference between metabolomics and the other omics concerns the many fields of study and application. Genomics and transcriptomics mostly concern research or medicine research, for example, and environmental research as well. But metabolomics is also very important in industry, in many industries, because metabolites are nutrients. We find them in food, and they are very important for nutrition. Metabolites also are drugs, so it would concern pharmacognosy and pharmacology. Many metabolites are also pollutants, so you will try to find them in the environment, and they are also very important in cosmetics. So beyond research, there is a lot of industrial application of metabolomics to characterize the small molecule content of a sample of food or cosmetic or any industrial or environmental sample. Here is a little update on the progression of metabolomics and how it developed as its own field of research. So if we look between 1999, which could be the birth of metabolomics as such, these graphs show the evolution every 2 years of the occurrence of a word or an expression, which is between brackets. For example, on the left, this is the word metabolomics in the database PubMed, which is a database for bibliography. And so you see that there is an exponential growth of the term metabolomics in the published science. If we look at the proportion of reviews, we see, on the contrary, that it has decreased. The proportion of reviews in the early age of metabolomics was around 45%. Almost half of the papers talking about metabolomics were reviews because they wanted to explain the principles and to make proof of concept. And now it's only about 15% because most papers mentioning metabolomics are original science, original research, just using metabolomics. So metabolomics has become more widespread, is used in many fields, and you don't have to explain so much now what it is and what it does. We can also look at the different tools that can be used, so a combination of the word metabolomics and either NMR or mass spectrometry. Well, we see that there is still both mass spectrometry and NMR that are very useful in metabolomics. Maybe we see an augmentation and increase in mass spectrometry occurrence. Mostly, I think, because many labs in the field of medical research have invested in mass spectrometry when before they were more at ease with NMR. NMR has been in hospitals for a very long time, especially through the MRI. So these are tools that were very well mastered by the people in this field of research. But now they found also that it was very interesting and complementary to use mass spectrometry as well. We can check also for the occurrence of gas chromatography and liquid chromatography. Up to 2016, we see little advantage for GCMS, but in fact, LCMS is increasing as well because it has a wider range of metabolites reachable with these techniques. And so we can look also by field of research, by crossing metabolomics and human diagnosis and drug. And we see that it has increased also over the same period. And this reflects also an increase of the medical sector. And then in plant biology, we can check for plant, Arabidopsis, and agriculture combined with metabolomics. Plant research was a pioneer field of metabolomics because, well, plants are very rich in metabolites of interest. But then many other fields of research cough up, and now plants represent 20% of the total number of occurrence in metabolomics. It's also interesting to see that Arabidopsis was something like 10% in the beginning, and now it has decreased, while some other keywords like agriculture have increased, or even industry, because we go from the more fundamental research now to more applied research using metabolomics. Now it's time for the second chapter, basic analytical chemistry. First, let's talk about extraction techniques, which are very upstream the workflow of metabolomic studies. The first step is to sample, take samples of the plants. So we do everything to quench the chemical and enzymatic reactions using mostly low temperature. So we use freezing techniques with liquid nitrogen. There is a lot of caution using this product to avoid injuries and also to avoid the destruction of the sample or its drying out. Well, you need to protect the sample from the liquid nitrogen. It's very interesting because it will flash freeze the sample. And so it will mostly freeze also any chemical or enzymatic reaction. Then the samples are stored at minus 80 degrees to avoid fast evolution of the metabolic content. Then there is a step of grinding. The idea is that the more fragmented is an object, the larger its surface. You have a curve on the bottom left with the radius of a sphere in abscess and the ratio of its surface over its volume. And you see that the smaller an object, the higher is its surface ratio to its volume. And so it means that the surface exchange for the metabolites to diffuse in a solvent will be higher. So, well, grinding can be done manually if you don't have too many, or by a Milburn grinder up to 8 samples simultaneously. So this is, of course, you do that with inoxidable steel balls that will be shaked and grind the samples. Now there is a concept very important in metabolomics, which is the polarity. It concerns the basic physico-chemical property of a molecule, whether it's a molecule of solvent or a molecule of metabolites you want to extract. So you will take advantage of the physico-chemical properties of these molecules to separate them from the matrix. So many of these metabolites have a specific set of chemical functions, like carboxylic acid, alcohol, phenyls, double bond, and so on. So an important physico-chemical property is the solubility of this molecule in different solvents, which largely depends on its polarity. So here are polar interactions. It's basically the ability of a molecule to develop hydrogen bonds with a polar solvent, the most polar solvent being water, but you also have alcohol, and non-polar solvent do not have heteroatoms like oxygen or nitrogen in their structure. They are just carbon and hydrogen, for example. They are not polar at all, and they cannot make these hydrogen bonds. So here you have the example of a water molecule that interact with each other through these hydrogen bonds. And this is the representation of a polar solute in water as compared to a non-polar solute in water. A polar solute will be surrounded by water molecules interacting with it and through these hydrogen bonds or weak bonds, sometimes ionic bonds. And this will disperse the molecule in the solution. But on the contrary, if the molecule is non-polar and you put it in a very polar solvent like water, there will be a thermodynamical favorable configuration, which is that these molecules cluster together, avoiding to increase the contact surface with water. So it's the idea that there is no mixing possible, there is no interaction, and so by default, these molecules will cluster together to avoid the contact, increasing the contact with water. Now this is a table indicating the different polarity of different solvents. So you go from up alkane, which is basically carbon and hydrogen. It's very non-polar at all. Then you have alcenes, halogenated, ether, esters, ketones, aldehydes, amides, amine, alcohol, and acids. Acid being much more polar because they are very, very prone to make hydrogen bonds and ionic bonds. We can measure objectively the polarity using the log P or X log P, which is a coefficient of partition of a molecule between two solvents of different polarity, water and octanol. So depending on how much of the molecule is soluble in water and how much is soluble in octanol, you can calculate a logarithm of this ratio that will be between -5 for very polar compound and +5 for very non-polar compounds. You also have for macromolecules the TPSA, topological surface area, in angstrom square. So this is a unit of surface, very small surface, but this will indicate what is the surface exposed and able to make some bonds with other polar molecules. It is topological polar surface area. There is a little mistake here. So there is an important database, which is PubChem. In PubChem, you can find a lot of information about many compounds. And well, it's quite reliable. You can use it to have some basic information like the structure of the molecule, but also its physicochemical properties, such as the X log P here. And this is a molecule of caffeoylquinic acid. It's a very common molecule in plants. And you can compare its polarity, which is -0.4, with the polarity of known compounds, like glucose, which is polyalcohol. So it's very polar, and it has a coefficient of -2.6. Phenylalanine, an amino acid, aromatic, -1.5. Morphine is an alkaloid with much fewer polar functions. It's 0.8. And palmitic acid, of course, it's got a carboxy function, which is very polar, but also a whole aliphatic chain, which is the CH, and no possibility for the hydrogens to interact with other molecules. So it has a very positive value. So you can see that caffeoylquinic in this configuration is somehow in mid-polarity range. So this is the classification of solvents according to their polarity. So the polarity increases when you go up, and you have an index of polarity, which is 1 for water on the top, 0.76 for methanol, 0.65 for ethanol or acetic acid, 0.55 for isopropanol. So you see that the four most used, commonly used polar solvents already have polarities that are very different. Water being by far the most polar solvent. And then you have apolar solvents like acetonitrile, dimethyl sulfoxide, butyl alcohol, dimethyl formamide, acetone, pyridine, chloroform, and then very apolar solvent like ethyl, like MTBE, toluene, cyclohexane, hexane, and pentane. You see, hexane is one of the less polar compound solvents available. Of course, you won't use this single criterion to choose your solvent. You will have to consider the danger of manipulating them, the price, and their purity. Or they must be LCMS grade to be used comfortably and avoid too many contamination of the sample by the solvent. And even by doing that, we empirically find many contaminants coming from the solvents. So here is a workflow of extraction and partial purification. Purification is something that can be added to the extraction. It can be total, you purify one compound, or just partial by just eliminating some undesired fractions of the metabolites contained in the sample. So you start with plant material. It's frozen, and of course you have organized your sampling. Then you extract them, you solubilize them in solvents, and stabilize them with low temperature, pH, polarity, depending on also you will increase solubility depending on the time of contact between the solvent and the sample, and the fact you will you will mix the mixture. Then you can have an optional step of purification. At least you will filter your samples to remove all solid debris that can come from cell walls and membrane and so on, and proteins. And then, well, you can use different techniques or a combination of them. You will have usually to separate the compound, identify them, and quantify them. So you will use chromatography mostly for separation, as we already said, identification through spectrometry and mass spectrometry or NMR, and quantification using different techniques with or without standardization. The choice of the techniques will depend on your equipment, but also on the physicochemical properties of the molecules. Especially LCMS or GCMS will be used depending on the size of the molecule and their ability to be volatilized. In red, I reported the different bias, the different problems you can encounter and that you will encounter if you do such analysis, like decompartmentalization, degradation, selectivity. Maybe the extraction is very good for a type of compound, but not very good for another. The degradation of the compound, some of them are very unstable. Think about ascorbate, which is vitamin C. It's very, very easy to degrade it because it is a very reducing compound and it can be oxidized easily. If you make a purification, it can be very complicated and very time consuming, and you will multiply the risk to degrade your compound. And then when it comes to the techniques, to the machinery and the techniques, then you will also have drawbacks regarding identification, dynamic range, and so on, that we already mentioned. So the objective, the goal of extraction is to quench metabolites, that will be shown in blue, then to solubilize the metabolite, extract them in a liquid, that will be shown in orange, and possibly to purify the metabolite, that will be shown in green. Sorry. So here I give an example of a very famous extraction technique, which is called the Blight and Dyer extraction, or methanol chloroform water, MCW. So first, of course, you harvest your tissues and you quench the activity in liquid nitrogen. You can also freeze dry the samples. It must be very well done to avoid degradation, but it's interesting because you lower the water activity by removing it, and so you will stabilize your product. Then you grind, of course, and this will increase the size, the surface to volume ratio of the plant particles, and you will conserve it at cold at minus 80 degrees. Then comes the extraction per se. So you wait a fixed amount of plant material and add methanol, including an internal standard. I will explain that later. So a certain volume you can agitate during 15 minutes at 70 degrees. So here the temperature is meant to degrade and precipitate the proteins, especially the enzymes. So it's a way to remove the proteins and to stabilize the sample by denaturing the proteins. Then you add chloroform, CH3Cl, and you agitate 5 minutes at 37 degrees. And the chloroform and the methanol are miscible. They will make just one phase. But then you will add water. And when you add water, the methanol and the water will mix up, but the chloroform won't mix with this mixture. So the chloroform will fall at the bottom of the tube because it is more dense, and the methanol and water phase will be on top. And the chloroform is a non-polar solvent, so it will trap all the non-polar compounds. This is why it's green, because all the chlorophyll, which are very non-polar, will be trapped in the chloroform. On the contrary, the methanol and water phase will trap the polar compound. And so depending on your goal, depending on the question you address, you can either analyze one of the phase or both of them. Once you've made the extraction, the extract must also be stored in good conditions, so in a place that is dry, without light, to avoid photodegradation, and of course at cold, at minus 80 degrees as well. There are many other techniques related to extraction. So I just showed a basic extraction, and you can of course use them depending on your goal and the type of sample and the goal of your analysis. I will just mention them, like filtration, ultrasounds, or microwave assisted extraction that will increase the rate of extraction. Headspace and SPME, which is solid phase microextraction for volatile compounds. Quechar methods for pesticides, and SPE, solid phase extraction, which are basically cartridge containing a resin on which to charge the sample and then push it with solvents to trap certain molecules on the resin and to elute the molecules of interest in a solvent. So I won't discuss that too much because we don't have time in this course. But then I will speak about a few other tricks used in extraction, like for conserving the extract, you can evaporate or concentrate the solvent. So it's interesting because if you have very low levels of the compound of interest in your sample, you may want to concentrate to avoid diluting it too much in a high volume of solvent. So you can use a rotavapor for this. Well, you have a flask here, the round shaped glass flask, which contains the extract. It is heated in a heat bath, and then the solvent evaporates and is trapped in a solvent trap by recondensation. In fact, there is a cooling system that will recondense the solvent vapors into liquid, and trapping it in another bottle on the left. And the system is rotating to homogenize the evaporation and avoid the cooking of the solid residues. Another technique very widespread is the speed vac system, where the samples there are in small amount, they are in microtubes, and they are placed in a heated vacuum chamber on a rotary plate in order to avoid the projection when the pressure is lowered. Then a pump is removing the air and lowering the pressure. This will increase the vapor phase transition, and the evaporated solvent is recondensed in a cold trap that is between the pump and the rotation system. So the extract can be as well concentrated or completely dried. This is interesting when you have a lot of samples, which is usually the case in metabolomic approaches. The rotavapor is more dedicated to pharmacognosy approaches. And then, of course, storage in cryoboxes with good identification for long term at minus 80 and for short term at minus 20. We have to mention the using of a QC sample, the quality control, that we also call the pool. The idea is to make a pool of all your samples. So you take a small amount of each sample and you gather it in what we call a pool or quality control. And you can use this to make many extraction tests. Then you can also make a pool of your samples. In this case, it's usually more called the quality control. And you can use it to check that your machine, your mass spectrometer, is not changing during the time of the analysis. Sometimes you have analytical runs that can last as long as a few days. And so in this interval, the parameters of the machine can change a little bit and the system will get dirty, and so the signal will change. And so the QC, the quality control pool, or the pool, is important to check that the machine is really stable during the analysis. So this is what is shown in this slide. The pool is used to develop analytical method, in particular to determine the mass of sample to volume of extraction solvent ratio, to experiment with different extraction methods. So you will use the same sample to test different methods, to adapt the chromatography, to explore the sample content. Maybe you want to know what is basically in your experiment, what metabolites you may find in your experiment, just qualitatively. And also to determine an internal standard. The internal standard is a molecule you will add to every sample, and that will track the small technical errors that you make. And you can correct the signal after by taking account the signal of this internal standard. It's a spy in your sample, if you want. But one condition is that your internal standard must be absent from the samples. Otherwise, there is a quantitative bias. And it must be added in the same amount in every sample. You can also voluntarily dirty the source of the mass spectrometer to stabilize the signal before injecting the sample. This is something we'll talk again later. And finally, you can correct the drift of the signal linked to the fouling of the source during the injection. This is what I said. The system might change during a batch of analysis, and so you can correct these changes by detecting it with the regular injection of the quality control. Let's explain now what is chromatography. The principle is to separate molecules from a mixture by differential interaction of these molecules with a stationary phase that we call also a resin or a column. In fact, it's a pack of resin inside a metal column. And a mobile phase, which is a gas or a solvent. So the important parameters to understand chromatography are the physico-chemical properties of the molecules, the physico-chemical properties of the stationary phase, the resin or the column, and the physico-chemical properties of the mobile phase, the gas or the solvent. In gas chromatography, we separate molecules according to their volatilization temperature. The different abbreviations we will find are GC, CPG, GCMS, GCFID. We use a capillary column and a flow of vector gas, which can be helium, hydrogen, or nitrogen. And it is very suitable for volatile compounds, mostly apolar compound or volatile organic compounds, but also for polar compounds after derivatization, which means after a chemical transformation that makes them volatile at lower temperatures. Liquid chromatography now means that we try to separate the molecules according to their polarity. So we use hydrophilic interactions, hydrogen bond, net charge, hydrophobic interactions. The abbreviation you will encounter are HPLC, LC, UPLC, LCUV, LCMS, and so on. The column in this case are filled with grafted silica microbeads, and the mobile phase is organic solvents, mixtures of water, methanol, acetonitrile, isopropanol, and it is suitable for bigger soluble compounds. Let's see the principle of liquid chromatography, and this is how the devices are connected together. So first, on the left, you have the mobile phase. These are bottles of solvents. And then you have a binary pump or a quaternary pump. Well, anyway, a pump that will pump these liquids into a circuit that is connected to the sampler. This is where we introduce the samples. Usually it's a small robot that is able to pump a bit of the sample and to introduce it in the circuit in which the solvent is going to flow. And then outside of the sampler, you have the column oven, which is controlling the temperature of the column. So the column is a metal tube filled with resin. So it contains the stationary phase. You will find C8, C18, helic. Well, you have different types of stationary phase, but they are always made of micro beads of silica, which are coated with different chemical function that will give its property, physical chemical property, to the stationary phase. And then the detector. So how does it work really, this column filled with stationary phase? So you have this metal tube, and inside, in orange, I represented the stationary phase. On the left, the injector. The sample is introduced into the circuit, which is flowed with a liquid of mobile phase. For example, a mixture of two solvents, water, which is very polar, and acetonitrile, which is much less polar. Then you introduce this sample, and these are the two molecules are represented, the blue molecule and the red molecule. And you start with only 5% of acetonitrile in the circuit. So you have a first graph on the left, which is the solvent gradient, which is the proportion of acetonitrile in the system, in the mixture, so that is going through the column, according to time. And the time is the chromatographic time. This is the time also in which we will report the elution of the different molecules. And then on the right, you have the detector. So what happens is that at the beginning, the molecules are mixed up in the head of the column. And then, after a while, the percentage of acetonitrile has increased. So we have a different solvent mixture. And this solvent mixture is suitable for the solubilization of the blue molecule. So now the blue molecule, which is the molecule number 1, is polar enough to be eluted at 40% acetonitrile, and it will interact less with the stationary phase, while the red molecule is still retained because it is less polar than the blue one, and it still strongly interacts with the stationary phase. And then at 60% acetonitrile, the first molecule reaches the detector, and the detector will register a signal in form of a peak. It is a chromatographic peak, and the top of the peak corresponds to the retention time of the molecule, which is the time the molecule, the first molecule, has been retained in the system, or also the time necessary to elute the molecule from the system. Now the red molecule is interacting more with the solvent than with the stationary phase, and it's moving into the column. And when you reach 80% of acetonitrile, it reaches the detector, and there is a second peak for the red molecule with a different retention time, a longer retention time, a higher retention time than the blue molecule. It means that it has been retained longer in the system because of its polarity. So here is, in fact, the principle of reverse phase chromatography. In this case, the most polar molecules are eluted first. It means that you need to increase the content of a non-polar solvent that will be a stronger solvent, with a stronger elution property that will solubilize the molecule. So here are the different types, the two main types of liquid chromatography for analytical purpose. On the left, you have normal phases. The normal phases are stationary phases made of pure silica or graphite with polar chemical functions like hydroxy or amine. These phases are thus polar. And so a non-polar solvent like hexane will be a weak solvent. It will have a weak power of elution. And when you will increase the polarity of the mixture, you will increase the strength of the mixture, the strength of eluting power. So in normal phases, non-polar solvents are weak solvents. They are not very efficient at eluting the molecules, while polar solvents are strong solvents with a strong elution power. And then you have reverse phases, which are made of apolar phases. So the stationary phase is made of apolar chemical function grafted onto silica, and we call them C8 or C18 because they contain 8 carbon or 18 carbon, for example. So these are aliphatic chains that will develop hydrophobic interactions with the molecules. And in this case, the weak solvent are the most polar. So this is exactly what I showed previously with 80% of water in the mixture. The solvent is weak and the molecule won't be eluted. And then when you increase the content of a non-polar solvent, it becomes stronger and it will elute the molecules. So, well, anyway, what you have to remember is that the idea is to go from a mixture of molecules to separated molecules with different retention times. And for this, we use a stationary phase and a mobile phase with different physicochemical properties. I've been talking about analytical liquid chromatography, so we call them analytical HPLC or UPLC. UPLC is just a modern version of HPLC. It is much more efficient for separation. The idea is to separate compounds from a complex mixture, like in a plant extract, in order to identify them and quantify them with the detector. And then you have preparative HPLC. The idea here is to purify the compound in a complex because you can add at the end of the column, you can add a recipient. And you can collect, after the detector, you can collect the flow. And the flow contains, well, the molecule that has been solubilized in it. So first you separate them, but in this case, you also collect the molecules after separation. And it's interesting because then you can test the biological properties of this molecule. Is it active on tumor cells, or is it active as an antioxidant, or and so on. You can also use separation to better identify the molecule by sending it to NMR for assessing the full raw formula and developed formula of the molecule. Now here is the gas chromatography, and this is the type of machines you will have. It is an oven that is thermostatically controlled, so you can change the temperature between 30 or 50 degrees up to 350 degrees Celsius. In this oven is placed a capillary column. So you see it's a thin, very, very thin column that is very long. It can be several meters long. And so it is. It makes these rolls of column. And on one hand, it is connected to the injector, in which a syringe can plunge to inject, for example, 1 microliter of the sample. And on the other side, you have the detector. So the column is fit to the detector. The sample is injected at high temperature so that the mixture is volatilized. It's volatilized. And this mixture in gas phase is pushed into the head of the column, which is at lower temperature to begin with. It's around maybe 50 degrees. So it will recondensate in the head of the column. And so I will explain just after how it works. Just look at how the column is made. So it is a coating that is very thermo-resistant, and inside there is a layer of silica and then a layer of stationary phase. But this is empty, okay? This is a tube, really. And in this tube, the gas, the vector gas, is going to flow. So here is how it works. So you have this tube, the capillary column, and the injector at high temperature is transforming the sample into gas phase, and it is pushing it at the head of the column, which is at lower temperature, T0, maybe 50 degrees, for example. And then the molecules are condensated on the coating of the column on the stationary phase. So they go back to liquid phase, adsorbed on the stationary phase. Imagine that there is a constant flow of helium, while the vector gas, that is pushing the molecule through this column. And then in the oven, you will increase the temperature. So the temperature inside the column will increase as well. And at a certain temperature, the blue molecule becomes more volatile. And then it's going to be pushed easily faster into the chromatographic system until it reaches the detector and makes a peak. Okay, so in this case, the retention time depends on the temperature of volatilization of this molecule. And then if you continue to increase temperature, you will release the second molecule that will be eluted and make another peak. So this is exactly the same principle. It's about separating mixed molecules and by knowing their retention time, trying to identify them, and also with the detectors. There are many, many different stationary phases. Some of them are polar, some of them are non-polar. So the choice will depend on the type of molecule under investigation. The selectivity, the power separation, longer columns have a higher power of separation. And the amount of sample injected, the higher you want an injection volume, the thicker must be the stationary phase. So this is one of the most currently used, frequently used column. This is a dB or HP5MS. And this is the coating inside. This is what is the stationary phase. It's methyl polysiloxane, so SE with methyl CH3. Many of them, 95% of the functions are methyl polysiloxane, and 5% of phenyl siloxane. In this case, instead of CH3, you have CH6H5. It's a nonpolar column. There is the MS, it's a 5MS, so 5 is for 5% phenyl, and MS means it has been treated in a way that there is a low bleeding. It means that it will be very resistant to high temperatures. Now, with gas chromatography, you can analyze many types of compounds, especially those with very low temperature of volatilization. So if the boiling point is below 320 degrees, which is roughly the maximum temperature you can expose the column before it starts to really degrade rapidly. So the volatilization temperature is lower depending on the polarity. The less polar, the lower is the temperature of volatilization, because molecules that do not interact with each other, because they are non-polar, are more easily volatilized than molecules that interact with each other, because you have to overcome the energy of interaction. And if the molecules are small also, they will be much more volatile. So gas chromatography is very suitable for analysis of gas, solvents, organic volatile compounds, hydrocarbons, terpenes, and lipids in general. But there is another way to analyze very polar compounds of small size after chemical derivatization. It means that you will transform all the chemical function with some polarity into chemical function that are non-polar. So in GCMS, you can analyze organic acids, amino acids, polyamines, fatty acids, simple sugar, oligosaccharides by transforming them before analysis, before injecting. So you can do that. One of the most current, frequently used derivatization techniques uses methoxyamine and trimethylsilyl agent. So you start with a polar compound with a low volatility. It means a high temperature of ebullition. Let's imagine a sugar. Okay, it might be a ketoexose or an aldoexose. And then first you will neutralize the carbonyl function, which means the aldehyde or the ketone function, with methoxym. So you will transform C double bond O into C double bond NOCH3. So instead of double bond O, which is quite polar, you will have OCH3, which is non-polar. And then the second step is to transform every proton that is able to interact, like here the alcohol protons, OH, but it also works with amines and thiols, SH, and so on, and carboxylic acid, COOH. You will change all these protons into trimethylsilide, SECH3 3 times. And by doing that, you will increase the volatility, so you will decrease the temperature of volatility, and you will decrease the polarity of the molecule. So it's a very interesting technique because you can analyze basic primary metabolites. And this is one of the most efficient methods to do so. Now, how can you quantitate a compound, quantify a compound by using chromatography? This is what we call peak integration. So here is a chromatogram. You have the retention time in abscissa, the absorbance, so the detector in this case is a spectrophotometer, and you set the wavelengths in the UV spectrum where most phenolic compounds absorb. So, for example, caffeoylquinic acid is your compound of interest here. You know that its maximum wavelength of absorption is 315 nanometers. So you will separate the compound by liquid chromatography, and if you know the retention time of the molecule, then you can detect where it is found, which peaks correspond to caffeoylquinic acid. So this is the peak you identified here. And now the quantity of caffeoylquinic acid is proportional to the area of the peak. So to the integral, mathematically it is the integral of the peak.